Buenas tardes a todas, todos y todes. Es un agrado saludarles. Mi nombre es Diego Salas Blujin y tengo el placer de presentar a la doctora.
Meet you, and I have the pleasure to introduce the Dr. Jenny Hsieh from Taiwan, who works in Puerto Varas. And she's a doctor in neurophysiology of the Cape Town University. Before, she worked in Bulk Sequencing Solution Cape Town, conducting research and development in sequencing. She is a researcher in NT-R&D genetics, developing enzymatic solutions for genetics market. I want to thank her participation in the Congress and also thank Kura Biotech for their sponsor on this event. I want to remind the attendees to the conference, please don't forget to leave your questions in the chat, because at the end we will answer them. And now, without further delay, we will begin the conference.
Hello, everybody. All right. Okay, so good afternoon, everybody. Oh, sorry, the crib notes are gone again, and I need those. Like, really need those. Sorry, the same thing is happening as before. I need to stop share. What is this happening? Okay, Zoom meeting, share screen, share that guy. Okay, is everything okay?
Yes, everything is okay.
Okay, and you can't see my crib notes. Cool, so sorry about that, and good afternoon, everybody. I hope everybody's had a good rest. When my boss asked whether I wanted to give this talk, I'd first thought about all of the things that I wish I'd known, about what else I could do with my degrees in science besides working in academia and at universities. And I definitely never thought that it would be possible that I'd be doing cutting edge science in Patagonia. I thought that if I'd known some of the things that I know now and how interesting my life could be, I would have been a lot less anxious as a student. So I thought I'd use my personal opinions, and so read very, very biased opinions. This is not scientific. And just use my story basically as an example, and use Kura as an example of a biotech company in Chile, not only because I work here and should probably say good things about my boss, but because I really believe in the vision of the company and bringing innovations from Patagonia to the world. So much so that I moved my family here from South Africa to be part of this very interesting project. So here I say for another time, and we'll repeat a couple more times, that these are very personal opinions, so very biased, and let the academics not skewer me. So one of the things that I wish I knew while I was studying was the difference between how and why science is done in academia compared to why and how it's done in industry. At the start, I really didn't want to work in industry at all. I love science, and I thought that industry was just full of greedy money makers who didn't care about science. But I needed to work because I needed money to finish paying for my PhD. And the decision to start working in industry would have been a lot easier if I had known these differences. So There are some myths that we have been told about the differences between academia and industry. Of course, if you're older than 15, then you know that firstly, nothing is ever pure. There's no such thing as pure learning or pure profit, but still we get the idea from media or the industries themselves, really, that academia is about pure learning, and you have this picture of this benevolent genius up in the ivory tower somewhere, and all you have to do is learn, and whatever you find, it doesn't matter. Science is the truth and it speaks for itself. And industry, on the other hand, has got this other image of evil scientists hiring evil geniuses with evil bosses hiring evil scientists. And if the evil scientist doesn't manage to do the job properly, the evil boss will then kill the evil scientist. And that's obviously not really how it goes. And the truth is that the aims between academia and industry are so different that, well, that is why the myths can't be true. So in academia, the aim is to understand and to communicate your findings. The aim is to publish so that other people can see what you've done, so that they can build on your work and so that they don't make the same mistakes, because that's a waste of time and a waste of money. The paper is the product, but that means that learning has, like, you can't learn anything that interests you. You have to learn what the publishers want to sell. In industry, the aim is to understand, sure, but also create. We learn how something works and interacts with something else, the same as in academia, but then you have to make or improve a product or something. The point is to, for example, make a COVID test, do something that can be done without, make a test that can be done without some expensive lab equipment, or improve the test so that it becomes more sensitive. That product that you make ends up being, well, the product. So let me think. Right. And another one of the main differences, obviously, is how you make the money, right? Pure learning and pure profit can't possibly be the case because of the way each sector makes money. Academia can't be pure learning because the way you make money is by writing grants. So you have to write grants to justify the various government bodies, or to various philanthropist groups to pay for the particular study you want to do. Or you have to join a particular study that has already been funded for this particular grant, with a particular grant, sorry. And these grants can be difficult to get depending on whether your interests match that of the government bodies that you're applying to, and who you know in the funding organization sometimes. Also, of course, academic grants might not be renewed if your studies don't achieve the goals you say you want it to, just because the science didn't turn out. In industry, on the other hand, as the company, we can't be pure profit, because if we choose pure profit and we hurt people, we'd end up hurting our own customers and we'd be shooting ourselves in the foot. So it wouldn't make sense for industry to be completely evil. And as the scientist in industry, once you're hired into the company, you're paid a salary. You don't have to justify for particular projects, really. You're by default funded for any of the projects that the company wants you to do. Of course, you can only do the projects that the company wants you to do. But sorry, but that's the kind of thing that you have to do for academia anyway, except in industry, we're honest about it. Obviously, to get a job in industry, it would depend on whether your experience is what the company is looking for at the time, and also who you know at the company, which is not that different from academia anyway. And if a project fails because of the science, you move on to another project and you still get paid your salary. Unless, of course, you did a bad job and then you get fired, and that's a completely different story. Okay. Here, let's see. Some career options or ladders of academia versus industry. So academia is more or less straightforward. After being a student, whether you're a bachelor's or master's or PhD, you can become a technician or lab manager, which is usually while you are studying for your next degree. If you're bachelor's, you're studying for your master's and whatever. If you've gotten your PhD, you can go on to do a postdoc, maybe 2 or 3, depending these days. And somehow you get promoted to an associate professor and then a full professor. And at each step, you're doing some sort of research, you're writing some sort of grants or applying for scholarships of some sort. You could be teaching some classes, or at the higher levels, you are picking up some masters and PhD students, and you start guiding those guys through. And at even higher levels, you start to join some kind of administrative communities and associations of neuroscience and those types of things, which is also generally all about communication or education. In industry, there is a much finer segregation in terms of the types of science that can get done. So in no particular order, I'm just going to go through some of these boxes, and the colors also don't make any sense. I've just made them different colors so that you can see the different marking borders, basically. So after being a student and coming out at any level, you can choose to work in the lab or not in the lab, or both. In the lab, you can choose to be a lab technician, lab manager, engineer. So the lab technician, for example, would be the hands. We need a lot of help doing quite a lot of experiments, and sometimes we just need people to do them. And lab technicians are very, very useful in that case. And generally, if you've got a bachelor's degree, you can be a lab tech. Lab managers, higher degrees, but you get to manage the laboratories, whether it's the R&D lab or all of the labs. That means you need to make sure that everything, everybody has what they need in order to do their jobs. You're checking that the equipments are actually working, that the pipettes are actually working, that the scientists aren't being idiots in the lab and all that. Then there is research and development. And this is what everybody thinks science is, but it really is only a small part of a small number of types of scientists to be in industry. And within here, you are thinking about new products, different ways to make something better, different ways to make whatever your company is trying to make. And you've got different levels of scientists. You can be a junior scientist where you're coming in with, say, a master's degree. And, then there are senior scientists, principal scientists, and team leads. There are levels with which to grow in the career as a research and development scientist. You've also got process development that takes the prototypes of the stuff that comes out of research and development, and they develop it into something that can actually be made with scale in manufacturing. All of these are departments that need scientists, whether it's to scale up certain products, figuring out how to make something more efficiently, manufacturing scientists actually meticulously making everything. And after the products have been made, it goes to quality control, which needs to check that everything has been made to spec. There are other scientists that actually validate the equipment. You'd be surprised at the number of equipment that needs to actually be fixed and calibrated and just kept in line in any lab. And depending on what kind of certification your lab goes through, you'll need different grades of validations. If you finish studying and you decide the lab is dumb and I really don't want to be in there anymore, or I'm super clumsy and all I do is break stuff in the lab, you can choose not to be in there. But we need scientists in sales anyway. We need scientists in marketing. How are you going to be able to explain and sell what a product is if you don't understand the science behind everything? Quality assurance. If you're a lot more into legal, into the certifications, it would be making sure that everything is actually ISO compliant, making sure everything we do is actually recorded properly so that there's evidence if anything goes wrong. You are the backbone of the company. You've got project managers that actually make sure that everybody does the thing and in some sort of coordination so that we're not bumping into each other and getting into each other's way. The CSO, the Chief Scientific Officer, well, he's in charge of all the science, right? The vision and everything comes through him. And bioinformatics, there's nobody that can do science anymore without bioinformatics these days. These days, you do a lot of experiments in silico, as in by computer first, to predict which experiments are even likely to work before you even do the experiment. And once you've done it, if you've put it into a sequencer or a mass spec, the data that comes out needs a bioinformatician to look at, because mere mortals can't understand anything that comes out of those computers. And if you want to be both in the lab and out of the lab, you've got customer support and application specialists. Here, you're working directly with the customers. Similar to sales, but the customer support tends to be post-sales. So sales guys, they will sell the thing to the customers, whereas after the customer has purchased the product, if they've got any questions, if they've got any problems, it comes to customer support. And you need scientists there to actually understand what the customer is doing, to be in the lab, to recreate the problems that the customer has, go out to the customer's labs and help them figure out right there what the problems are. And usually they're using the product wrong and they haven't read the instructions, but we need scientists to be there to help our customers use the things properly. Training is a huge part of their job, some like external for customers, but also internal for our own new recruits. And of course, reagent and stock purchasing and management. You'd think that's boring or you don't need a scientist for that, but of course you do. If you don't know the difference between D or L glucose, like buying the wrong thing can really make a huge difference in terms of what the scientists end up getting in the lab at the end of the day. So, and obviously there are many other non-scientists that are involved in the company, and I can talk about them later, but it's just, there's a huge number of ways that you can be a scientist when you work in industry. But of course, academia versus industry is a complete false dichotomy. It's not like there are only two ways to be a scientist. There is still, and these are also obviously small examples, but there's publishing and writing. People need editors for the scientific journals that you're trying to publish in. Scientific journalism, just to translate science that is happening to lay people in fast media and also popular science in books. So translating science, but in slower media and in much more depth. If you've ever read Steven Pinker, he can change the way you understand the world just because of the way he writes his books. And you don't need to be a scientist to be able to read those books and understand, but you do need to be a scientist to be able to write those books. And of course, government. I don't know all of the options available in Chile, but there are for sure a lot of options that you can choose from, like the ISP and Corfo, and I'm sure the Department of Health or Department of Science and Technology. There must be a bunch of places where government bodies need scientists. So there, again, lots of ways to be scientists. And as I said, I'm just going to be using Kura Biotech as one of the examples of a biotech company in Chile. And so for the rest of the time, I'll be talking about where we are, who we are, what we do, and why we do it. The why is easy. We want to give more scientists around the world the best tools so that they can use their success to improve the world. And we want to be part of the solution. Where we are is Puerto Varas. Besides the map there on the top left, these are pictures that have been taken by me or other colleagues around the office. On a clear day, you can see all the volcanoes. I don't know if you can see my mouse, but top right, bottom left, you can see the volcanoes literally out of the lab windows or the office windows. On cloudy days, you won't be able to see anything, but at least you'll be able to see rainbows, entire full double rainbows. And these views outside of the windows are actually one of the reasons why I moved to Puerto Varas. When you're having a really bad day and you have to make buffers that you really don't feel like pHing, it helps to be able to look at something nice outside. It just makes everything better. This building that's right in the middle, it's actually already we've already extended it. This is an older picture, and we're more than doubling the size of the building again now. So we're getting even bigger. And besides Puerto Varas, we're really all over the world. We have our labs and we're headquartered here, but we've also got offices in the States. And we provide many, many countries around the world with our products. Who we are? On the left here, where everybody's wearing yellow, that was a picture taken last year, just pre-COVID. And on the right, top right corner this picture was taken last week, literally. We're more than 60 people now. Could have taken us all out for a walk in Antiyanka. And when COVID started, you can see Hsieh there. It's the first time that she put on one of those hazard suits. We thought she looked pretty funny. She did a little dance and a pose. We have a lot of fun. Sometimes we work in the lab. Sometimes we make children work in the lab. And sometimes we just look at rainbows there on the left-hand side. It looks like we don't work at all. But yeah, this is who we are. The boys right there at the bottom, barbecue is very, very important, and they make amazing meat when we have barbecues. So I tried to find pictures that are representative of the vibe at work. Yes, there's serious work happening, but there is also everything else that is really just as important. And a little bit about what we do. So we've got 3 branches of the company. Finden deals with more of the toxicology products, and I'll talk about each of them a little bit as we go along. Avenir deals and sells more of the genetics related products, and Bleecker sells more of the proteomics related products. And I'm in the genetics part. Yeah. So about toxicology, again, this isn't my project, but I know enough about it to be able to introduce it to you. Beta-glucuronidase was actually Kura's first big success. This is what Kura was already making before I joined, and that we'd actually found that the beta-glucuronidase in the guts of red abalone was the fastest that anyone had ever found. And we used the leftover gut waste of abalone to actually extract beta-glucuronidase. That's the enzyme in the blue box there that comes after your gut. So basically, when you take a drug, it goes through your system, and a lot of your gut microbiome, sorry, Freudian slip, gut microbiome helps to convert some of the drugs using beta-glucuronidase into something basically that your body can break down. But we use this enzyme, we sell this enzyme to companies like LabCorp and Quest that test people's urine to see whether they have taken drugs, illegal or otherwise. Unfortunately, using the waste from the abalone industry makes it really difficult to control the quality of the enzyme that we get out of them. So we moved to making recombinant versions of beta-glucuronidase so that we can control the quality and the amount of enzyme that we make a little bit better. And over the last 4 years, we've actually become the producers of the best glucuronidase available in the world. I'm not just saying that because I work here, but LabCorp and Quest in the USA can buy any kind of beta-gluc they want from anybody, but they choose to buy ours because we are the best ones. For the genetics part, firstly, this is not our company. Illumina is an example of a sequencing by synthesis company. And also, sequencing by synthesis is not the only way to do DNA sequencing, but Illumina is just the largest genetic sequencing company in the world. More than 90% of anybody who does genetic sequencing uses Illumina machines. And genetic sequencing is probably the key, or at least a major step to understanding almost anything we want to know about biology. But these machines can only read short pieces of DNA at a time, like 75 to 300 base pairs, while organisms and animals have millions or billions of base pairs in theirs and our genomes. So you have to prepare DNA in very specific ways that will firstly allow the machine to read it, but also depending on what kind of information you want to get out of your sample, you also have to prepare your sample a different way before you put it into the machine so that you can get the right kind of data out. I'm not going to go through every one of these possible ways to prepare the DNA or RNA. You would have to make the RNA into DNA before it can be read on the sequencer. The point is that at every single step, there is an enzyme that is needed to make every change to the RNA or DNA. The analogy that I like to give people is that if DNA was paper, we would be making the stationery. So, for example, if you're looking second from the right. If we want to take genomic DNA from the top and cut it into small pieces, we would be using endonucleases, which will be like molecular scissors. And after we've cut them into pieces, we want to stick adapters, which are known DNA sequences that can tell the machine details about the piece of DNA that it's reading. And we'd need ligase, which would be like the glue that would stick the adapter onto the fragment of DNA that we've just cut. And since there are usually very few copies of these fragments, we might need a copy machine to make more of each of the same fragments, so that it'll be easier for the sequencer to read. So polymerase would be equivalent to our photocopier, for example. And these are the tools that we scientists would be making to manipulate just DNA. Those enzymes are products. And similarly, if you want to sequence proteins, and again, LCMS is not the only way to sequence proteins, but proteins also need to be processed in very specific ways for machines like the mass spec to be able to read what they are. They have to be prepared a particular way in order for it to be vaporized in any case, and then ionized and shot through this tube. And at the end, you have this profile of peaks that give the computer, basically, that the computers then figure out what kind of peptides and proteins were in there in order for you to sequence your particular protein sample. And here again, there are a lot of different ways for you to prepare your proteins, and between every step is an enzyme. These protocols, whether you're talking about the DNA ones or the protein ones, can take you anywhere from, the DNA ones can be shorter, they can be about an hour, but they can take up to 20 hours or 3 days in some cases, depending on what kind of data you want to get out. And part of our job is to find enzymes that can do it faster, or find some kind of process or solution that'll be able to make it just a lot easier for scientists to do. There are some scientists that, imagine if your experiment took you 3 days. It's a mission, right? We work on trying to make our lives just a little bit better. So this was what we were doing before COVID-19 disaster, whatever you want to call it. We watched from January in 2020, thinking that people in China and Europe really need to get their act together. In February, we were in a bit of denial and hoped that it would still just get under control. But by March, we realized that the situation was really just totally out of control. And we asked ourselves whether we could do something to be a part of the solution. Of course, we all had projects geared towards making tools for various forms of sample preparation for DNA or proteins, like I'd said before. And making diagnostic kits or detection kits was never a goal for our company. It wasn't why any of us were actually hired, really. But when we asked ourselves what we could do to help, we realized that between all of us, we actually had enough skills and previous experience to be able to make COVID kits. So the first problem was that there was a worldwide shortage of test kits. That is why we chose to go this route. And at the beginning, we couldn't even test enough to see how bad the spread of infections were, to even get an accurate count of numbers. The Chilean government worked hard and made some real gains in purchasing kits from China, but that was still not enough. And we decided to make some RT-PCR kits for official lab tests to try and relieve the problem of international supply of these kits, and just be able to help in some way. Of course, there are still supply shortages now for all sorts of testing. We're not, we can't do everything, but we can be part of helping. But we also knew that the official qPCR tests take too long for the results to get out to people. Companies, schools, communities all need to know who has COVID so that only the people who have COVID can be quarantined completely. Otherwise, even essential services won't be able to function. We all knew that the most time-consuming part, we also knew, sorry, that the most time-consuming part of the whole testing process was RNA extraction. So we made it easy so that we didn't depend entirely on the official testing system to get society going again. So instead of the normal column extraction that can take an hour, we did something that took 8 minutes. And we made it really easy so that you didn't need a whole lab to do it. You mix 3 ingredients together for the blink master mix. You add a bit of saliva, mix it up, and you leave it for 5 minutes. And then you heat it, well, basically put it in boiling water for 3 minutes. And that's your extraction. That's the whole hour gets saved, basically. And we also changed the detection method. So if you take a drop of the mixture that you just extracted and you put it into our resist kit, which is just a mixture of 2 things together, in 30 minutes, you have an answer. We made a color version so that you can read a negative positive for pink versus yellow. And we have a fluorescent version as well, so that if you want to read it on a machine, you can. This is one of the, basically we won in some Chile Innovation Award for that. I forgot to put a picture in there. Sorry, Javier. Javier is my boss. He won the innovation for leading this particular project. But this kit is so simple that you can literally do it in the back of a cart, which is what we did to pilot everything. We built our own little, it's not really a lab, right? It's just a cart with some UV lights and some benches. We put in some pipettes. There's a small centrifuge in there, 2 water baths, one for the heating and the other one for the resist temperature. And that's it. We have bottles to collect saliva in, and that was just how we got started to pilot everything. And obviously, there's a whole bunch of scientists that were in the field to make this work. We started in Frutillar. Claudia was really brave and went out and did everything with, sometimes she had team members there in Chilean, in Chonchi, in Valdivia, depending on where there were problems and needed additional testing that couldn't be done. We went out both to test our own kits, but also to do what we could for the community. And as our CEO likes to say, there are some people who are alive now because of the things that we did. Of course, the scientist doesn't just make COVID test kits in a few months just because we sat around the table and said we would. It takes a whole team of scientists and non-scientists to pull together all parts of the job. So besides the various roles of scientists that I'd spoken about earlier, our whole team is useless without all of the other non-scientists who actually make up the bulk of the numbers of people, of the number of employees. Logistics helps to bring all of our reagents in. I mean, without finance, nobody gets paid. Human resources, obviously, we kept needing to bring new people in because we were making products. Cleaning staff, thank goodness for the cleaning staff. And marketing. So marketing, you have scientists who do marketing, and you've got non-scientists who do marketing. You'll notice sometimes that scientists don't make the things look as pretty as they need to be. So both types of marketing people are very necessary. In the end, really, I just think that biotechnology is one of the most powerful tools that we have in our hands, and our heads to make the world a better place for everybody. And there are many ways to be a scientist, whether you're in industry or not. Could have made ourselves part of the solution, even though we can't solve everything, but we can each make ourselves part of the team that forms part of the solution. And we go on, right? We empower scientists with better enzymes, best enzymes, hopefully, so that we can each improve the world in our own way. And that's it. Science is a verb.
Muchas gracias.
Thank you so much, Jennifer, for this very nice presentation, which well allows us as biochemists not only to see that the academic area is there, but there is also the industrial area. We can expand and broaden our view of how this can also work, or how science can also contribute directly to the industry. Or for example, in your case, regarding these COVID kits, which I found to be really interesting. Now we are going to proceed to a Q&A section, and we do have a question from Kevin Guzman, who is asking, okay,
Decide to sell in the south of Chile.
Sorry, can you repeat the question?
why did you decide to sell in the south of Chile? That's the
To
question.
sell? To sell or settle?
Settle.
Ah, okay.
To sell in the south of Chile.
Because Kura Biotech invited me. They had it was a they at the time, it's a we now, but 2.5 years ago, basically, the original idea was to build out our genetics product line. And I have a lot of experience with genetic sequencing kits and how to make those. I had a lot of that in South Africa. So I did about 4 years worth of sciencing in that field. And they were needing those people, so I applied. Also, there's a way of living here that I'd wanted to do in South Africa. We have a piece of land, we have a piece of mountain in South Africa, and I wish I could live out in nature and still do science, but that's not an option in South Africa. If you want to do science, you have to live in a big city. But Kura is this really strange place where you're out in a tiny little town, and I can live out in nature, but still do science. It was just the best of both worlds. So now I have a piece of forest here instead of a mountain, and I still get to do science.
Me imagino que es una experiencia bonita vivir en.
This must be such a beautiful experience to be so close to nature and continue doing science and the first level science. We have a question from Ignacio Vega, who is asking, have you shared your experience with other Chilean companies? I presume he's talking about whether you have made some connections maybe with the rest of the Chilean industry or other Chilean companies regarding your experience in Kura Biotech.
Not yet. I've been quite busy here.
Pero como Kura Biotech,
No, but as Kura Biotech, as a company, I'm not sure if the company has done something in that regard. I'm not sure if Kura Biotech, yes, if it's Kura Biotech has been contributing, for example, to the Chilean industry or to Chilean companies.
Contributing in what way?
En resolver problemas, puede ser.
Maybe solving problems in the agricultural or livestock industry, for example, something like that.
All right. Understood. Yes. So we do have some projects along those lines. So we have had some salmon testing, testing for Pisci rickettsia, for example, for the salmon industry. They have come to us with some questions. We've developed some solutions, but yeah, we'll see how those go. But yes, we would like to be as involved in Chile as possible. I mean, obviously, this is our first stop because it's closest, but we've already sold to the rest of the world as well. So wherever people wants to use our solutions, we'll be here for them.
Y bueno, y una pregunta un poco
And there is a similar question. Have you also had any relationships or connections to Chilean universities in the country? Yes, for example, have you worked in any projects, or has it only been Kura Biotech on its own and that's it?
Me personally?
No, Kura Biotech as a company.
Yes, no, Kura Biotech has a lot of connections with a lot of the universities. A lot of our validations for our COVID kits, for example, went through a lot of our, basically everybody comes from some university here in Chile, besides me and Hsieh, I guess. So we've all got connections at universities, and our friends there definitely help us out with a lot of stuff. For proteomics, for example, we have a lot of connections there to try. I can't tell you exactly which universities because I'll also say it all wrong. So, but yes.
Y un poco...
and a little bit going back to this COVID detection kit. Is it already validated by the Chilean Health Service, or is it used both at the national level and internationally? Is it also used in other countries as well?
It is definitely already, both of them have been approved by the ISP in Chile. For international, I'm not sure whether we've sold through, our aim is to sell to the rest of LATAM, at least. The US doesn't really need us. So we're looking to selling to the rest of LATAM as well. The last I heard, we're headed into Bolivia, but it's not really, I don't know the marketing as much. I'm just the scientist.
Está bien, está bien.
It's okay, don't worry. And Rocio Lorca is asking, do you accept students for an internship, for example, or who are about to do their thesis at Kura Biotech? Do you accept undergraduate students?
Yes, we do, and yes, we have. One of our employees now actually was interning by us before, and she did so well and enjoyed it with us. So when she applied, we said, hell yeah. So yes, we do. It doesn't pay much, but we do.
Ahí hay una oportunidad
There is an opportunity to get started then.
Yes.
I'm not sure if we have more questions. Okay, here is a question. Why did you decide to start working on the industry side of for sciences, but from your personal point of view, what caught your attention and made you say, okay, I'd rather focus on applied sciences in the industry?
Because I wanted to be a little bit closer to the results of what I was researching. So as a neuroscientist, I was looking at things that was going to take 20 years to even end up in a textbook, not to mention in some kind of medication or something that people would use. Whereas in industry, when you do something, you make something, you can see your results a lot more quickly. So that's one. The other part was just, I thought it was a lot more honest. I got tired of trying to write grants and ask for money. I felt that when I was writing grants, I was making promises about how I would change the world. This project that I do is going to do this and solve methamphetamine issues and no psychosis in South Africa ever again. But you knew that that was never going to happen, right? You do the study for 3 years, somebody pays you, and then you're going to be like, oh, further research is required. And that's just our way of asking for more money. And I didn't want to keep doing that. I felt that I could make just as much of a contribution as a scientist without needing to constantly justify my salary and just be paid a salary.
Me parece una buena reflexión.
I believe that to be a good reflection. Yes. Once again, thank you so much for your presentation. It was fantastic. It was, I think, for many of us, allow them to see a different view of what a scientist can do in different areas, which they may not be considering for now, but are just as important and can make changes. And once again, thank you so much. And also thank you so much, Kura Biotech, for your contributions to this Congress, and also for allowing us to have this lecture.
Of course, you're very welcome. Thank you.